Welcome to Population and Ecosystems Investigation 9, Genetic Variation. In this investigation, students investigate the underlying mechanisms of change in population by breeding imaginary animals called larkies. They learn how organisms inherit traits from their parents and how dominant and recessive alleles interact to produce traits in a population. This investigation is comprised of 4 parts. Part 1, Variation of Features. Students observe the variation in human traits and larky traits. Part 2, Larky Genetics. This lecture discussion introduces students to the structures and mechanisms of heredity. Part 3, Breeding Larkies. Students use letter tiles to simulate the 4-gene genotype of larkies. Part 4, Punnett squares. Students learn how to use Punnett squares to predict the probability of offspring traits when the genotypes of the parents are known. As always, behind each investigation tab is the matrix that outline the goals and standards as well as step-by-step instructions for how to teach the unit. Part 1, Variation of Features. Review feature, variation, and traits with students. Ask students if they think there is variation among humans. Tell them that they will be looking at 4 features of their body. Give students 3 minutes to determine their trait for the 4 features. Poll the class using Transparency 20, Features and Traits, or draw the chart on the board. Point out the 4 features and under them the 2 traits associated with each. Start the polling process. Discuss the results using the scripted questions in the teacher's guide and summarize by confirming that there is variation in human populations and that variation is what makes every individual different from everybody else. Introduce the larkies by projecting Transparency 21. Tell students a larky is a little make-believe dog-like rodent that we will be working with to help us understand the big ideas of genetics. Identify the 4 features of the larkies. Tell students to look on pages 56 and 57 and choose a larky that they will be working with. Return to Transparency 20, poll the students, and record the information for the larky traits. Part 2 of Investigation 9 is primarily lecture discussion. I recommend that you take the time out and jot down some notes about this discussion prior to beginning this investigation. You might even want to print up your notes so that you can distribute them to students to follow along as you go, as well as the transparencies that you will be projecting. Make sure that students have copies of them so they can make connections and distinctions as you go along. Spend some time on gene locations, just pointing out that each feature is controlled by 1 gene and each gene is on a different chromosome. Notice that some of the alleles are labeled with uppercase letters and some are labeled with lowercase letters, but both upper and lowercase letters are for the same feature. Introduce genotype by telling students that the alleles are the code that determines the traits of the larky. The combination of alleles in an organism's chromosome is the organism's genotype. Tell students that alleles don't all have equal influence in determining traits. Some alleles have more influence. These are called dominant alleles. They are represented by uppercase letters. Less influential alleles are called recessive alleles, and they are represented by a lowercase letter. Review the gene locations chart, identifying whether an allele is dominant or recessive. Model for students how to decode genotype. Give them a copy of Transparency 26, From Genotype to Phenotype. Bring their attention to the 3 columns at the bottom of the page. Discuss the information in each. Model for them how to transcribe the genotype to find the phenotype. Once they have completed, have them complete the drawing of the larky based on the translation of the code. Introduce phenotype to students. Tell them that the way an organism looks is its phenotype. And every organism has its unique genotype composed of paired alleles on paired chromosomes. Those genes produce unique traits in those organisms, and the traits that the genotype produces results in the organism's phenotype. Have students review the vocabulary by giving them a genetics vocabulary sheet. Give them 10 minutes to fill in the blanks with the new vocabulary. Allow them to work together to make sure that they agree on the vocabulary. Assess students' progress by reviewing their student sheet. Assign the reading Mendel to Human Genome, solving the heredity puzzle by dividing the reading into sections and grouping students with each section, having them read and summarize the section and sharing out the main ideas with the class. I do this because this is a very large article and we can lose students' focus really easily if we try to sit and read the entire thing. Breeding Larkies Part 3 is very detailed and very complex. It is very important that you watch the video on breeding larkies as well as having students watch the video. Because if they do not follow the procedures precisely, the simulation will not work. Begin by using Transparency 28. Larky parents tell students they will start with the genotype of the two parents. Describe the traits using the Larky genetics table. Introduce recording with Larky ID cards. Tell students that if a gene has a dominant and a recessive allele, record the dominant allele first. Point out the symbols for male and females and make sure that students know they are printed on the card. Discuss recording the generations. Tell students that the P generation is always the parent. First generation offsprings, or F1, filial, means son and daughter, and that subscript numbers are used to indicate the filial generations. Follow the script in the teacher's guide to complete the following steps. Introduce breeding using Transparency 27. Again, making sure students view the video clip of Larky breeding as well as using the student notebook for Larky breeding steps. Take the time to discuss the Larky breeding record. Identify the second row has 3 Larky IDs. The first 2 are the F1 offspring of the P generation, and the third will be a migrant from another team used to breed the female. We will not be breeding brother and sister. Introduce homozygous and heterozygous. After students notice that both alleles are the same for each gene in both parents, tell them that there is a scientific term for a genotype that has identical alleles. It is called homozygous. The prefix homo means the same, and zygous refers to the genes. Homo may be 2 dominant alleles or 2 recessive alleles. If one dominant and one is recessive, it is heterozygous for that gene. The prefix hetero means different. Continue the breeding process following the instructions in the teacher's guide. Have students spend time with the multimedia as a do now or wrap up. There are 2 simulations that can help students understand the genetic concepts of genotype and phenotype. Assess students' progress by having them use the response sheet, genetic variation, to check their understanding of the basic principles of inheritance. Part 4 introduces the Punnett square. Talk students through the process of using a Punnett square. See the script in the teacher's guide. Have students spend more time working on Punnett squares in the multimedia section, as do now as a wrap-up. Assess student progress using the Punnett square sheet. Also using the genetic variation practice sheet. Assess students' understanding of the entire investigation by giving them mid-summative exam number 9. You can give students more practice time on Punnett squares using a Bikini Bottom genetics practice sheet located here. This concludes investigation number 9.